From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on May 16th, 2024. Hello, Micronauts, and welcome once more to today's Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 41st episode of Matters Microbial. I can hardly believe it. Those of you who watch and/or listen and spread the good microbial word to others, many, many thanks. It has been suggested to me that we go immediately to our podcast guests without information about my classes and students. This is Microbe TV, not Mark TV, I know. So we'll give that strategy a try. Hopefully we can get more views and listens as a result. Fingers and toes crossed. I really enjoy this podcast and hope I can continue to share it with you. I promise that I will let you all know if any particular #OMG, overwhelming microbial greatness, or #WTM, what the microbe, results occur in my lab or courses. Why are we made up of many small cells instead of a few large ones? One issue is the volume of a cell versus its surface area, as you can see. The volume of a cell increases much faster than its surface area, and that is a problem. After all, food needs to come across that membrane, as do waste materials in the opposite direction. At the same time, the word microbe seems to suggest smallness, as in this diagram. You can see that there are protists like amoebae that can get quite large. There are also very small microbes, so microbe can be a funny word. But remember, generally speaking, we can't see microbes without a microscope. But as usual, microbes have interesting adaptations and strategies. For example, here is an image of Thiomargarita membimbiensis next to a fruit fly head. Or Thiomargarita magnifica, a true mammoth among microbes, next to a dime. And of course, the subject of today's podcast, improperly pronounced Epulopiscium. See how it compares in size to the protist paramecium and even an E. coli cell. Now, I teach about this topic every time I have a microbiology class because of how the subject of cell size integrates to so many concepts. I simply love discussing this topic with students, so it's genuinely an honor to welcome a true luminary to this field. Dr. Esther Angert, Senior Associate Dean and Professor of Microbiology in Cornell University's College of Agriculture and Life Sciences, to the podcast. Welcome to our Quality Quorum for today, Esther. I've been in so many seminar rooms watching you give talks over the years, but this is the first time we've met. It really is a privilege.
Oh, Mark, I'm so happy to be here. Thank you for inviting me to tell you a little bit about our work.
Oh, I so enjoy it. I have to, and as I say, I teach about this stuff every semester, so I want you to hold me responsible for any misstatements that I make.
I like your introduction so far. It was wonderful.
Well, that's kind of you to say. I think before we say anything else, did you want to say a few words about how you entered microbiology? Because I know it's quite a story.
Sure. It's how much time do we have? um, I have always loved the natural world. My earliest memories are of running around in fields of grass and mucking around in a swamp that was behind my house where I grew up, and coming back and having to get hosed off because of all the mud. And so I love the natural world. I love thinking of it as an ecosystem and all the interactions that happen in that microbial or beyond world. And so I've always loved symbiosis. I've always loved plants, and I've always asked questions. How did that happen? Why does it look this way? Why do all of that type of tree look the same? And so I've always had that curiosity about the natural world. And whenever I went to college, I very naturally gravitated towards being a biology major. During my freshman year, I had work study, and so I looked for places that I could do biology or learn a little bit more about biology, and I was very fortunate to get a job in a micro prep lab. And so I would help out making all the media. Yeah, absolutely. Making the plates, learning about the microbes in a very superficial way, because I had to, go into the classroom and write the names of each microbe and misspell it on the board and all of the media.
Esther, how many petri dishes can you carry at once? That's an important question.
Oh, uh, I can put my hands around a stack of 6.
Well, many times students make fun of me for kind of clipping the top with I'll walk around with 50 or 60. It's really kind of fun.
Yeah, yeah. No, no, I usually use trays, and so I'm not quite that.
Much smarter.
Did you play violin? Maybe it's a.
Oh, good Lord, no. But it's interesting that you had that introduction early on, and especially the ecological construct to it, because that's one of the things about your work with giant microbes that I think people don't appreciate, because it actually has a genesis in ecology.
Absolutely,
And if you
absolutely.
think about it, hasn't that been the case for every one of the giant microbes that people study? I
You are
can't.
absolutely right.
Mm-hmm. And
And the sorry, I'm going to talk over you a little bit.
that's okay.
One of the common themes to all giant microbes so far, I'm putting that out as a challenge to future micronauts. So far, we can't grow any of the giant microbes in pure culture in the laboratory. So everything we do has an ecological context, we have to go and learn about those organisms in their natural environments.
No, I think that that's really important. It's so common for people who are new to microbiology to think if you just find the right media, you can grow anything. And I'm a little nervous about that. I'm not saying it's wrong. I've been proved wrong many times. I don't have ego involvement in that. But I believe that many microbes are in concert with other microbes. We see lots of evidence for that. Chlorochromatum aggregatum is a great one and fascinating, a subject for another day. But what I'm getting at is organisms have to work together.
Mhm.
So I think that you have a beautiful image of some giant microbes that have been seen. Can you talk about them a little bit?
Sure. We've known about giant microbes for more than 100 years. And if you go back into the literature, you see descriptions of things like Spirochaeta plicatulus and other giant unknown, never named microbes that have been pulled out of all sorts of environments, like the gut of tadpoles and anaerobic muds. And they all sort of fit a model. They were giant, they were hundreds of microns in length, but only a few microns in diameter. They were all really skinny. As we moved towards, more contemporary times, we started seeing microbes, seeing things like Epilopycium that seemed to fly in the face of this idea that, as you so nicely introduced, that surface area is important, and so these long, skinny, incredibly long microbes all have a very thin cytoplasm, so that no part of the cytoplasm is very far away from the external environment. And moreover, they're rod shaped or spiral shaped, so that they have a lot of surface area to encounter nutrients in the environment and exchange waste products. What really flew in the face of that was Epilopycium. Epilopycium can be 600 microns long, and it can be 80 microns thick instead of just a few microns thick. So the question that posed to scientists, the question was, how does this microorganism get away with not being skinny? And then Thiomargarita came along, Thiomargarita namibiensis, this enormous It can get to be a millimeter in diameter. Spherical cell, a millimeter in diameter. How does it do it? Well, it's got a trick. And the trick is that 98% of the cytoplasm is not really living. 98% of the cell volume is filled up with a fluid-filled vacuole. And the active cytoplasm is found in a very thin layer just under the cytoplasmic membrane that's only a few microns thick. And so it kind of fit in this model of being a skinny cell without being a skinny cell.
In other words, it's cheating.
It's cheating.
That's what I
It's a
tell my
bubble.
students. Yeah, it's really interesting. This idea of this huge vacuole, which to my knowledge has nitrate in it to act as an alternative electron acceptor. But again, that super tiny cytoplasm, as opposed to epulopicium. Bit by bit,
Yay.
I'll get there, which is just enormous. And I want to emphasize again to listeners and viewers that there are very few microbes that you can see with the naked eye, and Epulopicium is just about there. About the size of a grain of table salt would be one way of looking at it. And the
Yeah.
idea of a single cell of that size is just remarkable. So where do you find this organism?
Before I get into that,
Oh, sorry.
let me just highlight one of my favorite images, and that's a tube of epilopycium cells. I don't know if your micronauts are familiar with microcentrifuge tubes, but I when we start purifying cells away from the environmental milieu that they're in, we do some settling of the cells in microcentrifuge tubes. And when we get them very enriched, I hope you can see that that's a tube full of little white rods. Each of those little white rods is an epilopycium cell. And so you can see them with the unaided eye. It's pretty
Fascinating.
remarkable.
Just amazing. And Thiomargarita magnifica, which is not really the subject today. The image that people have seen of it that I mentioned before, next to some dimes. I have many thoughts on this I'll share with you another time, because I think there's relevance to what you're going to be talking about, which is what I call, we'll come back to this, service to the cytoplasm, which sounds like a motto, and it kind of is. How are these organisms motile?
Oh my goodness. They are highly modal. So
You have a
one
video to show.
yes, please show that video. One of the contrasting features between Thiomargarita and Epilopycium is its liveliness. So when people started looking at epulos, they saw these wriggling masses of highly motile cells that were in the gut of these surgeonfish. Compare and contrast that with Thiomargarita, which is a very low metabolic rate. It's just kind of hanging out. It's not motile. It's just going with the flow. Reproductive-wise, these cells, they're epilopycium's packed full of ribosomes. It's reproducing once every 24 hours, and it could be producing 2 or as many as 12 internal offspring during that 24-hour period. That's pretty darn good for a bacterium that's living in the gut. Thiomargarita, much slower. I don't know how frequently it divides, but it certainly isn't every 24 hours.
What
So
is
that liveliness
yeah,
is
what's the
another.
mode?
Oh,
How is it doing it?
they swim. They swim with bacterial flagella. And so that was one of the early mysteries when people started looking at Epilopycium under the microscope. They're saying, look at this. Oh, it's big. It has to be a protozoan. But where are the cilia? We can't see any cilia, and there's no flagella. Well, that's weird. It wasn't until people started doing thin sections that they saw, oh, there are filaments on the surface. And guess what? They're the size of bacterial flagella.
But a lot of them.
A lot of them. The surface is covered with bacterial flagella. It's kind of like a shag rug.
I think an obvious question, Esther, is what's in it for an organism to be large like this? I mean, what were the size has got to work against it in some way. So there has to be some wonderful advantage to being this large. Do you have any thoughts on that?
I think you'd need to get small, like an epilopycium, and go into the gut of a fish. It is a highly competitive environment. It is packed full of ciliates and flagellates and probably viruses and all sorts of guys that are out to get you. There is a ciliate called Balantidium jocularum that it also lives in the gut of Nasotonganus, and it will eat epulos. So I think one of the advantages of being big is that not too many things can eat you. So it's a survival thing.
I've heard that about size to avoid predation. Interesting.
Absolutely. The other thing that I think is important for Epilopycium is that it's got a ton of methyl accepting chemotaxis proteins. So, methyl accepting chemotaxis proteins are important for a bacterium to sense its environment and move either towards a nutrient, towards something that it wants to take it up, or move away from things that it doesn't want to encounter. Epulos have a ton of MCPs. When you open up the gut of a surgeonfish and you unravel it, because they're highly coiled, you unravel it, you find epulos in a very narrow region about midway along the length of the intestinal tract. I think that they're probably sensing their environment and staying in the right environment in the intestine. And that's going to be important, with all those flagella, with all the MCPs. But if you're big like that, you can move fast. So epulos are some of the fastest known bacteria. They can travel a lot of distance. And I think that they can probably make their way against peristalsis and everything that's happening in the gut, and they maintain their position in the right environment.
This makes me wonder, have people haven't seen these in ruminants, have they?
No, we haven't.
Well, it's clearly time to introduce them, right?
Yes.
So, related to this, what is the advantage of being small? So these are the pressures. I mean, I know that there are things called ultramicrobacteria. That's a creative name, but there are certainly some bacteria that are quite small. I don't mean the ones that respond to starvation by essentially shrink wrapping their central dogma machinery, but they're basically very, very small. I mean, thinking about Pelagibacter, for example.
What's the advantage of being small? Is again that high surface area to volume, and something like Pelagibacter that's out in the middle of the open ocean, which is a low nutrient environment, having that high surface area compared to your volume increases the proportional number of encounters with nutrients that are going to be important, essential for you to survive in that low nutrient environment.
I,
Epulos, high nutrient environment.
it's funny whenever I hear these kinds of things, part of the back of my head says, the microbes don't care what you think, Mark. They're going to do what's best for them.
Mm-hmm.
And that's like, I need a feeling for the organism, to borrow the line about Barbara McClintock, right?
Yes.
Such a fascinating subject. So you do get these large microbes,
Yes,
and they're in very specific kinds of environments, and they have to
absolutely.
deal with this surface volume problem. Now you can
Yes.
get the occasional cheaters. That one species of Thiomargarita is kind of cheating. So tell us more about Epulopicium.
Sure. So Epilopycium, we've been studying it for a long time, and the foundation of our studies has been what's different about Epilopycium. At first, we thought, okay, well, maybe it has a really weird genome. Way back when we first really started studying it, and we didn't know where it fit in the phylogenetic tree, we thought maybe it was a novel form of life. And then it turned out to be right in among the gram-positive bacteria, like the Clostridia. And so it seemed like, well, it's not on a weird long branch of the tree. What's its trick? What's the way that it has modified its cell biology to allow it to get to be so big? One of the striking features of the cell was that it contains a ton of DNA. So if we were to compare a large epilopycium cell With a human cell, a large epilopysium cell would contain maybe 250 picograms of DNA, whereas a human diploid cell contains 6 picograms of DNA. So that was the first clue, but it also opened up possibilities. Is it an enormous genome with unusual functions? We kind of didn't think so based on its evolutionary history. So a couple other ideas came to mind, and one was that maybe it's got lots and lots of genome copies of a typical sized bacterial genome, or maybe it's got a lot of copies, but portions of the genome have been amplified that are required to support its metabolism. Long story short, we've generated data both from quantitative PCR, and also now that we have a fairly good high quality genome, we've found that Epilopycium viviparus, our model organism, has about a 3.28 megabase genome, which is right where most bacterial genomes are, the size. But it's there in tens of thousands to hundreds of thousands of copies, depending on the size of the cell.
Can I break in and say that there's an image that I hope appears now that shows after staining with a dye DAPI that stains DNA, you can see that there are lots and lots of, and I'm going to use this word, nucleoids. And what I mean by that, maybe we just call them genomes, but there they are. And they tend to be at the top, at the border, rather than distributed throughout the cytoplasm, is the last I heard. Is that still true?
That's absolutely true.
Now, there are lots of reasons for that I hope that you and I can brainstorm about. Excuse me, like you haven't thought about it and know all about it. Excuse me, that I can ask about. How's that?
That's
Um,
wonderful.
but but it's fabulous, and I have so many questions about it. But you can see that they tend to appear at the edges of the organism. But if you change the focal plane, so you look over the surface, you can see them all there. As many, in some cases, I've heard, as you mentioned, 30,000 copies. And that's just fascinating. So where does this organism normally live that it would be so strange?
Epilopycium, I like to talk about our model organism, Epilopycium viviparus, but it's just a representative of a diverse group of bacteria that I like to call the Epilo family. And they're all inhabitants of the intestinal tract of certain species of surgeonfish. So surgeonfish are tropical, marine, coral reef associated fish. There are about 80 known species. And the species that we study the most is a really beautiful fish called Nasotonganus. And Nasotonganus, all all surgeonfish are very interesting looking. They might be kind of dull in their color. And so whenever you're swimming around, maybe snorkeling on a coral reef, you might not notice them unless you look closely and you see that they might have projections off of their nose, and they have spines on their tails, and that's where they get their name from because somebody thought they were like a surgeon's lancet. Some of them are brightly colored, some are yellow, some are movie stars like Dory, but surgeonfish are very diverse. Diverse family of fish. Some of them eat a lot of zooplankton. Some of them eat a lot of invertebrates, but others eat detritus. Others eat invertebrates and detritus or some combination with algae. And some are strict herbivores, and so they're eating primarily algae, either brown algae, green algae, red algae. And depending on the fish species, they have a preference for what they like to eat. We find that these epulos, these giant bacteria from maybe 10 microns up to the 600 microns, we find that they tend to be in the herbivorous fish in the surgeonfish family, and some of the detritivores. So right there, you're getting a sense of what these bacteria might be doing. It's diet-based. And now that we have genome sequences, at least from Epilopycium viviparus, we see that this organism is really capable of breaking down the polysaccharides that are available to it in the algae that the fish is eating. The fish can't break down these unusual polysaccharides, and that's what Epulo is doing for them. They are breaking down those polysaccharides, they're fermenting them into short-chain fatty acids, which the fish can then use as an energy source. They also probably are producing, I know that they're very biosynthetically capable. They're probably producing vitamins that the fish can use, and maybe even amino acids that the fish might be using.
Well, this is really fascinating because we've spoken several times on the podcast about how the microbiome can have all kinds of effects in a number of different systems, including you and me. And here's an interesting idea. Also, it suggests to me, feel free to correct me, that this is a very high nutrient area. And because it's high nutrient area, that might help a little bit with the nutritional needs of such a large organism.
I totally agree with you. If we look at all giant bacteria, they're all highly polyploid. And to make DNA, you need a lot of carbon, you need a lot of energy. And so, one common theme is that giant microbes are found in places with rich carbon, or that they can access carbon readily. So, with Thiomargarita, they're mostly autotrophs, and so they're taking in CO2 and fixing it, and they've got a great energy source because they're carrying around their nitrate with them in a little satchel. So they're in an energy and nutrient rich environment.
We call that a,
Epulos
we call that a nitrate scuba tank in my classes.
are that's perfect.
Yeah.
Epulos are also in a very carbon rich environment. They're living in the gut of this fish. The fish is eating on a regular cycle. It eats during the day, it sleeps at night, it gets up, it eats again. And the epulos have coordinated their life cycle along with that pattern of pulses of nutrients, and then maybe times where it's a little bit more lean, and then the next day there's another pulse of nutrients.
I think this is really cool. But there's got to be a limit having to do with this. And though this isn't really cheating, maybe we'll call this a little bit of a fib. Is that I ask students a question and I'll say, how big are your lungs? And then they'll say that their lungs are basically this size, but not when you unfold them. It's an issue of surface area, isn't it? So I explain, like a piece of paper, you can fold it up to be very, very small, but actually the surface area can be very large. Is it not true that Epulopicium does that itself?
You're absolutely spot on correct. Whenever you look at a thin section, an electron micrograph of a thin section through the very top surface of an epilopycium cell, you'll see this unusual cell envelope that I'm not going to talk about. But then just under the surface, you see this highly infolded membrane. And we think that that's key. It took us a very long time to make progress on figuring out how and what might be accommodated by this, but certainly at the most superficial level, it's a higher surface area. With the Epilopycium viviparous genome, we went on to take advantage of this life cycle that I just referred to. Because epulos are so synchronized with the behavior of their host, they coordinate their life cycle with the availability of nutrients in the gut. And so we have naturally synchronized populations with respect to offspring development. I didn't talk much about offspring development yet. Should I do that now?
Well, I mean, I think the species name gives it away, but please. I just you know what's funny is when I teach this subject, and Esther, I have to tell you, this is such a I'm sorry for fanboying, but this is such a big deal to me, and thank you again for being here, because when I teach about this, it's amazing to me. When I do, I want the students to focus on all those invaginations, those infoldings. I compare them to mitochondria, to the lungs, to certain nitrogen oxidizers that do that with their membrane, again, because they need all the extra surface area, thylakoids and in cyanobacteria. But they get focused on what you're going to talk about now, which is the way this organism reproduces.
Yeah. So it's unusual. If you think about bacterial or lots of microbes, how they reproduce, we think about the baby cell. And the baby cell is really happy and it's growing to about twice its starting size. It's replicating its genetic material, it's making more stuff, right? And so it gets to about twice its starting size and then it divides in two using binary fission. Epilopysium doesn't use binary fission. Instead, the largest epilopysium cells that we know of divide near the poles of the mother cell, if I can use that term. They divide near the poles, and some of that massive amount of DNA that's in these cells is trapped in each of those polar cells. Those polar cells are then fully engulfed by the mother cell. So the mother cell membrane migrates around each of those polar cells and then meets on the far side, pinching off that polar cell in a membrane-bound compartment within the cytoplasm of the mother cell. It then starts to grow. It grows until it almost completely fills the mother cell cytoplasm, and then it bursts. The mother cell envelope splits, and the offspring cells emerge from the mother cell, leaving a dead husk behind. So we could take advantage of that and look at gene expression at different stages of that developmental process just by going out, collecting a fish, sacrificing it, and collecting the epilose in its gut at different times of the day, representing different stages of that offspring development.
But you have to admit it looks like live bearing.
Oh, yes.
And
Yes.
and that's what the image that you're looking at now shows. And this is the thing that completely fixates my students is they'll see that and it's like they can't look away.
Yeah.
And like they'll forget the other things I'd like them to remember, which because you're going to be telling me some things that's going to thread that needle pretty well, I think. Because we'll be
Yeah.
able to talk about viviparity, which it isn't really, but it kind of looks like it, and your work with gene expression during this process. So I'm really excited. I'm taking notes.
Oh, very good. So early on, whenever I started working with epilose and seeing this really weird production of bacterial babies, um, uh, we started asking questions. Well, where did this come from? It doesn't make sense, except in the context of phylogeny. So one of the closest relatives of Epilopycium is a very cool bacterium, also that can't be grown in culture. It's a gut microbe called Metabacterium polyspora. It's a multiple endospore former. And when you look at Metabacterium early on in that spore formation process, it looks a heck of a lot like what's happening with Epilopycium. Except for these giant epilose never take that last step to make a phase bright, dehydrated, fully resistant endospore. So vivipary, it's more like sporulation that's just not really completed.
Yeah. I love the fact that this is something that you find in a coral reef. Where are you going to collect them, by the way?
I've gone primarily to an island off the northeast coast of Australia called Lizard Island. There's a research station there. It's been running since the 70s. It's primarily designed for people who are looking at fish and looking at coral and studying the ecosystem. And I and my intrepid students have gone there as microbiologists and tried to do some microbiology there, which is challenging but not impossible.
Oh, it's wonderful. I love to ask people that question that do the kind of work that you do, because I remember when I was in graduate school, please forgive this story, and Charlie Yanofsky was a professor there, and Paul Ehrlich, an ecologist, was a professor there. And Paul Ehrlich would consistently tweak microbiologists during seminars. He would say, he had this booming deep voice, and he would talk like this. And he would say, here is a typical microbiologist, and there's Charlie peering into a fermenter. And he goes, and here is my laboratory. And he shows the Rocky Mountains. And it's true that most of us are looking at Petri dishes, or not really all the time, but you know what I'm getting at. But you get to go look at things in truly remarkable, beautiful places and find such beautiful, remarkable organisms there to study. It's a win-win.
I've been so fortunate in my life, and I think fortune favors the open mind. I'm going to say in this instance, is that I've always been very open to new opportunities and not fixed on one particular path in my life, and that has led me to some really amazing and exotic places.
That's wonderful. So now we know that they live in the gut of herbivorous fish.
Hmm.
Okay. We know that they have 30,000 genomic equivalents in nucleoids. I have many thoughts on this. I'll stop myself. We know they're highly modal. They're helping the fish. They reproduce in this remarkable fashion, which looks unique, but isn't. It's a kind of a side path, an evolutionary side path to typical sporulation. I'm so happy. And now you're talking about trying to learn what genes are expressed during the process of, I'm going to still call it viviparity.
Yeah. Yes. So we've looked at the genome content and we've looked at gene expression and we've compared it to another bacterium that we can grow in culture that's a close relative, that's a spore former. It's called Cellulosyllaticum lenticellum. It's a cellulose degrading bacterium, and it was first uh, isolated from anaerobic muds downstream of a paper mill. Not terribly exciting, but it's a very nice organism. We see that it makes phase bright spores. It only produces one spore, but we know that it has a complete sporulation pathway, and we have a complete genome of it. So we compared the genome of Cellulosyllaticum lenticellum, focused in on the genes that are only used for sporulation. So we know that there are processes that are important to produce a spore, but they're also important for vegetative growth. We ignored those genes involved in those processes and just looked at the genes that we know are core to sporulation, and then looked with the simple question of, are they there in the Epilopycium genome? The surprising thing is that, yeah, lots of them are. The only things that were missing, well, you could probably predict what was missing. It's those last few important stages of sporulation where the cell becomes mineralized.
Mhm.
So spores become mineralized because they pump dipicolinic acid and calcium into the spore core, and it gets dehydrated. If we look at Epilopycium, there's something like, don't quote me on this, maybe 9 genes that are important for synthesizing dipicolinic acid in the mother cell and then transporting it into the developing spore. In Epilopycium, I think there are only 2 of those 9 genes that are present, whereas Cellulosyllaticum lenticellum has all 9. So it's missing that ability to make a dehydrated spore. It can't go back. But all of the other features of sporulation, all of the genes required for engulfment of the forespore, for laying down that special peptidoglycan layer called the cortex that's important for that dehydration step, all the cortex genes are there. All of the genes that are required for synchronizing and coordinating the gene expression that has to happen independently in the mother cell and the forespore and with the right timing, the sporulation sigma factors. SPO0A, the master regulator of sporulation, Epilopycium has all of those, and they're expressed. And they're expressed at the right time. So we have very good evidence that this process evolved from endospore formation.
So I do have to say, when you say they're being expressed at the right time, I want everyone to understand you mean that 30,000 copies of these are being expressed at the right time.
As far as we know,
Yeah. Well, I know you're being very cautious and I appreciate that. But just to give you kind of a heads up for later, please don't respond to this now. what keeps those different genome equivalents the same, because I could see so many opportunities, for lack of a better term, for cheating. And of course it's hard to study, but you certainly have the situation where you took what I'm going to call structural genomics, the because you can't really do experiments on the organism directly. So you were able to fish these genes out. You were able to show where those genes were expressed in this organism, which I'm just going to call undomesticated. And this really drives home a point that I want everyone to remember. I've talked about, like, the three really awful things, the three deadly heresies, and one of them, or the deadly centrisms, I should say, the deadly centrisms. And the last one is coli centrism, the idea that everything acts like E. coli, when in fact only E. coli acts like E. coli. And one of the things that people do is they focus on model systems, and then they think that that reflects to the whole natural world. And of course it does not. And so what's deeply cool about what you're doing is you're taking an organism. I have this image of you like a lion tamer with Epulopicium on the other side, and you're still getting good information out of it. When most other people might kind of say, well, I could do this in Salmonella, which is just a version of E. coli. And you're going to have all those people saying that, when the fact is the really cool information, and I'm not putting down the original stuff. I just love the cool, strange borderlands of microbiology. So, Esther, I'm a fan.
Oh, thank you. I'm a fan of Epulopicium, and so I'm going to deflect that and just say
Please.
focus on this extreme organism, because even though people who work on model organisms might say, "Oh, you'll never make any progress looking at this organism. Why should we even think about it?" It's because I feel like those extremes are where we learn key bits about cell biology that have to be extreme in this organism and may be so subtle that you don't notice them in E. coli or Bacillus subtilis.
Mhm.
And so, sure, we're working with one hand tied behind our back because we can't do genetics, but we can do microscopy. We can do transcriptomics. We can do proteomics. We can do genomics. And comparative genomics, which has been super powerful. I don't want to totally dismodel organisms, because if we didn't have that foundation of Bacillus subtilis, how would I know what genes were used in sporulation or offspring formation in the epulo group? So
No, of
I
course. Yeah.
totally appreciate that, but I also think we got to get out there and look at diversity, because you're absolutely right. What's true for E. coli is true for E. coli.
And I recognize the reference. I'm going to have a student make a painting of that. I already have the elephant in E. coli. That's a Jacques Monod quote. What's true for elephants is true for E. coli. It's very elegant in French, which I don't speak, but it needs to be changed. What's true for E. coli is true for E. coli.
Yes.
And again, just as you said, I want to be very clear. I am not dissing work with E. coli at all. It's a big petri dish we're on, this planet Earth. And there's lots of room for people to use different approaches and different things of interest. And my biggest worry has been that people would get too directed. And that's why I like it when people make these jumps into unusual and strange organisms, because they give us more perspective. For example, if you look at Thiomargarita magnifica, they have these things that are calling pepines now, which are very similar to your multiple genomes. Now, I'm not asking you to comment on that, but you see the paradigm shifting. People are getting new ideas from this.
Absolutely. And I think that I like to think of it as convergence, where the form of Thiomargarita, all Thiomargarita have a big fluid-filled vacuole, no matter their size, no matter their shape. And that presses that active part of the cytoplasm right under the cytoplasmic membrane, where it's getting that input from the environment and it's responding in a timely manner because it's right there. Um, so I feel like epulos have converged on that by having their nucleoids closely associated with the cell membrane, so that response times can be fast. And even though these are two very different organisms in two very different parts of the phylogenetic tree, they've converged on the same solution. They have very different metabolisms. They have some similarities in their reproduction that I'm not going to go into, but they've converged on the same theme to try to get past that, what we thought was a physical barrier, that surface to volume ratio that was keeping microbes small. Sorry about the anthropomorphism, but
No.
making microbes stay small.
It's interesting. I've often said that the hand of Darwin is on all that lives, and it's absolutely true. There are only so many solutions, and sometimes the different solutions to a problem are the most interesting ones. Again, to me. Which is interesting, if I may ask, if you imagine the cell of Epulopicium, see, eventually, if I write it down and tattoo it in my brain, I'll get it correct. Um, when you have the nucleoids around the periphery of the cell, how thick through is the cell? Because they don't have nucleoids in the center.
That's right. And so that's a bit of a puzzle, and people ask me that all the time. Well, what's the rest of the cytoplasm doing? If the active cytoplasm is right under the surface of the membrane, what's all the space about? And I don't have a great explanation. I know that it's packed full of ribosomes. I also would speculate that it's the really big cells that get really puffy and fat. And I think the really big cells that are puffy and fat are full of offspring. And so that space
Oh.
also accommodates the growth of those offspring cells in a very fast time.
Now that's really interesting because if you think about the Min genes for cell division, there's that whole nucleoid exclusion idea where you just have stuff in there and that helps establish where the new copies of the genome are going to be.
Mhm.
So that's a very, very interesting approach. And I used to talk to Elio Schechter about this quite a bit, the OG, definitely the OG, and
Yes.
and original germ, I know. Uh, Elio is a hero. I mean, you want to talk about microbial heroes, but one of the things that he and I talked about once is this idea there needs to be service to the cytoplasm. The idea that you're going to be transcribing a gene, you're going to be translating it, and you need to get it to a particular place. And so now you're dealing with diffusion issues, right? And so because of the needs of such an enormous cell, you have many different genomes. Now, that does lead to a problem of what maintains constancy between the different genomes. And I believe in achromatium, there's evidence that some of them are starting to diverge in a single cell from one another. I don't know if people have done that in epulopicium.
Yes, we've done some studies to look at the diversity of genes in Epilopycium. And so one of the things that, if we go back to the life cycle, that asymmetric division that occurs early in offspring development is a bottleneck. It traps probably, this is kind of a guess just based on fluorescence, it probably traps maybe 100 copies of the chromosome in that polar cell. And then it grows. We don't see any evidence of DNA passing into an offspring cell after it's engulfed.
Okay.
So it's going through a bottleneck all the time. We did a study looking at population diversity because if you see that bottleneck happening and you know that these are strict anaerobes, they don't like an oxygen-rich environment. And that's an assumption, but I think a pretty good one. They don't produce an endospore. How do they get from one fish to another? And the short answer is that juvenile fish eat feces of adult fish. I know.
No,
um,
cows, same thing with cows, right?
that's right.
Yeah.
That's right. Um, so these organisms are kind of going through a couple different bottlenecks where
Mhm.
population diversity might be reduced. So we looked at just a few genes from individual cells, looking at a population within the fish, and also looking beyond a fish into additional populations of epulos in different fish, in different individual fish, but the same species. And we found that the populations are actually pretty darn diverse. And what we think is happening, again, this is a model, hard to test in the gut of a fish, but what we think is happening is they have all the components to be competent cells and to take up DNA from their environment. And when we look at the diversity of those genetic markers in individuals, we see that there's reshuffling going on. And so knowing that bacteria like Vibrio cholerae can take in 10 unlinked pieces of DNA and recombine them into the genome, we think that epulos are maintaining their genomic diversity, their population diversity, by reshuffling using congression, this ability to take in unlinked markers and stitch them into your genome to rearrange things. So we think that they have that innate ability to maintain population diversity. What happens inside of the cell? There could be mutations occurring, right? The offspring is still growing and replicating its genetic material, and single point mutations can happen. You could repair them, but you could accumulate mutations. Even deleterious mutations, but because of all of those genome copies, maybe it's not having a big impact on that individual. So there are ways that it can maintain population diversity through recombination. In fact, we see that when we do some fancy analyses that recombination is super important to maintain population diversity, much more so than mutations. I think I'm digressing a little bit.
No, because it's really it's fascinating to me. But the whole point is this is how it's dealing with many copies of the same genome, and this is not. It's not quite the same, but you see many examples of organisms that are polyploid. I mean, bacteria that are polyploid. So these strategies are out there in the pool of available strategies that microbes can use, which is fascinating to
Absolutely.
me.
We have an unpublished study where we do some very deep sequencing on individual epilopycium cells. And for the most part, we're seeing that the genome copies are all identical, although we're seeing a few potential SNPs that are a proportion of the population. So I think they're probably maintaining very identical copies within an individual cell, maybe because of these bottlenecks, but also because of the really good repair mechanisms it has.
This is just the greatest. I've been quite enthusiastic, and we are running a little bit short on time.
Oh,
No, that's not your fault at all. I was going crazy again, like I do, you know. But you know what? I've said many times that I'm a giant goofball, and I am. But my goal is to be microbiology's giant goofball. And what you've told us today is an amazing story. Do you have any final words for our listeners and viewers about this wonderful system that you study?
I do. I do have some inspiration, or at least it's inspiration for me. Epilopycium was first identified in the 80s. I know it was the previous century and all, but I think that it's incredibly fascinating that with Epilopycium, with Thiomargarita namibiensis, with Thiomargarita magnifica, we're still discovering these amazing, huge microorganisms that aren't that micro, right? We can see them with the naked eye. And if we're just scratching the surface of that one trait of being a giant. Just imagine the diversity of organisms that we still have to discover and learn more from.
It's what makes microbiology such a wonderful field for everyone to keep track of, to study. It is indeed a microbial planet. Esther, it has been such a joy talking with you. Thank you so much for your time, and especially your patience while we figured out how to get things working on our end and your end.
Thank you so much for your patience with me,
Oh,
too.
not at
This has
all.
been a
This is
pleasure.
oh my goodness. I hope to see you in real life at some point. And I want to tell you, yes, I noticed the tardigrade behind you, of course. If you look behind me, I can't begin to count the number of tardigrades that are back there, but that's what happens with me. But what I was going to say is I want to wish you and your research crew and your family the very, very best. And to remind everyone that I'm going to be putting up some links, including the wonderful visuals on Esther's research group website. Thank you so much for your time.
Thank you, Mark.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with awesome links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Esther Angert is Senior Associate Dean and Professor of Microbiology in Cornell University's College of Agriculture and Life Sciences. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I always enjoy that. I hope that you've all enjoyed being part of our quality quorum today. See you next time on Matters Microbial.